So it is my great pleasure to introduce Dr. Emma Meaburn to give today's Centre for Educational Neuroscience seminar. Emma is a reader in human genetics at Birkbeck, University of London. After completing her PhD in behaviour genetics at King's College London, Emma joined Birkbeck at the Centre for Brain and Cognitive Development in 2010. Her research investigates how genetic variation shapes individual differences in behavior and the social and biological processes that mediate these influences. Emma is also a member of the Centre for Educational Neuroscience, where she works with colleagues to better understand the real-world challenges facing education and how genomic insights might help. Emma recently completed a secondment at Nuffield Foundation, where she led the research and development of ultimately a report entitled Genomics, Neuroscience and Education. She'll be talking to us today, or you can see the title, Genes, Learning and the Classroom Insights and Implications from Educational Genomics. Over to you, Emma.
Thank you. So can you hear me? Yes, perfect. I'm going to minimize everyone because otherwise I will just stare at my own face for the next 40 to 45 minutes. So yeah, if there are any issues, please shout or raise a hand. Thank you for the lovely introduction, Michael, and for the invitation to speak today. I'm Emma. I'm a behaviour geneticist, as Michael said, at Birkbeck, and a member of the Centre for Educational Neuroscience. And the field of educational neuroscience, or science of learning, aims to understand how learning works in the brain. So the question I'm kind of hoping to tackle today is, has genomics shed any light on this? Or perhaps more importantly, has it helped us understand the causes and correlates of individual differences in educational outcomes or conditions or difficulties that impact learning? So my goal today, somewhat punchy, is to try and give you a kind of high-level overview of what we know about the role of genes in education and learning, with illustrations from my own research and a focus on polygenic indices. So I'm going to try and unpack what they are and what they can do, and just as importantly, what their limitations are. And I'm going to illustrate one of the ways in which they can be useful, I think, as a research tool to unpack kind of the untangle what is the kind of real world phenomena of gene environment correlation, using a recent study led by my PhD student Nisa Rainey. I've opted largely to focus on the research perspective because I'm hoping this will set the stage perfectly for Michael Thomas, who's going to be speaking in a couple of weeks about the potential translational uses of genomic data and polygenic indices in education, I believe. So hopefully it sets the stage for him there. So again, as Michael mentioned, I'm an academic at Birkbeck University in the Department of Psychological Sciences. I'm a member of SEN, but I also recently completed an 18-month research secondment to the Nuffield Foundation and the Nuffield Council on Bioethics. So there I undertook a visiting senior researcher position, and my task was to examine the implications of advances in neuroscience and genomics for understanding differences in learning in children, particularly thinking about what the implications might be for policy, for educational practice, and the ethical issues that arise not only through the basic research, but also thinking further ahead, the potential translational use of genomics in education. So I've weaved some of those kind of insights throughout my talk, but I'm not really presenting those reports here. If you're interested in reading them, the QR code should take you to the webpage, and they're free to you should be able to just download them using that QR code. So before we dive into the research, I want to make sure we're all on the same page with a few key concepts. I'd imagine not everyone here is a geneticist, so this is my kind of super quick genomics 101. So genomics is a study of the genome, an organism's complete set of DNA, and my focus is on humans. So the complete set of the 3 billion A's, G's, C's, and T's that make up the human genome. And so as the slide says, our genome contains all the instructions we need for building our brain and our body. And we inherit one copy from our mother and one copy from our father. So my little homegrown individual here I've sketched out in the nucleus of every nucleated cell in their body is a copy of the genome. They inherit one copy from their mother and one copy from their father. And I'm just showing a short stretch of the genome here, a short stretch of the DNA sequence. So the genome is comprised of these 4-letter alphabets: adenine, cytosine, thymine, and guanine. And it's the specific sequence of these letters that guides everything our cells do. So the DNA sequence is very, very important. And about 99.6% of the DNA sequence is identical between any two unrelated people. So for the most part here, looking here at my little figures, there are quite a few places where the DNA sequence doesn't vary. But importantly, that tiny fraction, that 0.4% of variation, actually relates to quite a lot of differences in the genetic code between individuals. So the genome's 3 billion base pairs, or 3 billion letters in length. So that means, roughly speaking, that your genetic code is going to differ from the person sitting next to you in about 12 million places. So for example, here in this position here, the DNA sequence can be an A or a G, or it can be a T. This individual has inherited 2 copies of the T. This individual has inherited 2 copies of the G. So these DNA differences, kind of bring us to the fundamental question that behavior geneticists such as myself are interested in, which is fundamentally, to what extent do these differences in our DNA sequence account for the differences we can see between people in how they think, feel, and act. There are broadly speaking, two kind of questions that geneticists such as myself tackle. The first question is how much? So how important are these DNA differences in accounting for differences we see between people? So this is really thinking about questions about heritability and also about prediction. And secondly, the how. So thinking about the mechanisms, again, thinking about science of learning here, understanding mechanisms and processes linking biology to learning. So how do these genetic variants work to influence how we think, feel, and act? What impacts are they having on the brain? Or actually, as I'm going to be talking about today, to what extent are these effects moderated or mediated by the environment? So you might be thinking that's all well and good, but what's the connection to the classroom? So for decades now, research using twin and family studies has given us a really clear answer. It's robust evidence now that traits such as educational achievement, or even how long you spend in education, which is typically called attainment in genomics, which can be very confusing, but these two things are heritable. So roughly speaking, when we're looking at these things, about 50% of the differences that we see between people in a population can be accounted for by differences in their DNA sequence. And this isn't just about educational achievement. Many other traits and conditions that we know have an impact on a child's learning and educational trajectories and experiences, like autism, like ADHD, like various mental health conditions, are also known to be highly heritable. I'm not wanting to negate the importance of the environment, of course. We know genes aren't the whole story. Heritability isn't 100% for these things. And we know that policies or interventions focused on the environment can have long-lasting impacts, especially for disadvantaged children. But fundamentally, we know it's both nature and nurture. And critically, they don't seem to be independent. So it's both genes and both the environment. They're not independent of each other. And so fundamentally, I think if we want to understand the causes and correlates of educational differences, then we shouldn't be ignoring one at the expense of the other. So just if you weren't convinced when I'm talking about genetic influences, I just wanted to show this slide here from a now quite famous study that was published in 2015 from Tinker Polderman and colleagues, who conducted a big meta-analysis of all the twin studies that have been conducted in the past 50 years. And pretty much no matter what domain you look at, you can see evidence of heritability. So nearly every aspect of human individual differences is heritable. So looking at this heritability here is this purple bar. Looking at kind of cognitive function, it's around about 50% for psychiatric conditions. And in fact, even when we're looking at environments, or what we're calling environments, we can see genetic influence. The kind of, I guess, the joke for this slide is that for 50 years of research, you find out it's about 50-50 in terms of genes and environment. A quick note on heritability, but I think, again, Michael Thomas is going to be talking about heritability in more detail in his seminar. But I just kind of wanted to highlight that heritability is just a statistic that's estimating the relative contribution of genetic influences to your trait in that specific population at that specific time. It speaks only to differences between people. So it's all about individual differences. It doesn't tell you anything about an individual. And it doesn't have a fixed or true value, and it will vary across contexts. It will vary across cultures, across countries, and across timeframes. But generally speaking, that can be quite useful, and pretty much when we're looking at these heritability estimates, they'll always kind of fall around about kind of 40 to 50%. So for a long time now, that's all we knew. We had statistical evidence from twin and adoption studies that genetic variation seems to play a role, but we didn't know what genetic variants and where. And again, really one of the questions we want to get at as a research field in behavior genetics is mechanisms. And so if we want to understand what's going on, then we need to identify what specific genetic variants are underpinning these heritable traits, where they're located in the genome, to ultimately try and understand how they work. Are they in genes? And if so, what are they doing in those genes, and how is that impacting development? So about 20 years ago today, not today, but about 20 years ago, roughly speaking, technology was developed called SNP microarrays, or you might have heard them called DNA arrays or B chips, that allowed researchers to systematically genotype genetic variants across the genome. So essentially, these arrays or B chips allow you to look at many spots across the genome where we know genetic variation resides. And so you can kind of look at all of these spots and see how they differ in your cases versus your controls. So this led to the development of this technology, these arrays that can look at upwards of 2 million common genetic variants in a single go, in large numbers of individuals and at scale and at speed, led to the development of, I guess, this major research paradigm called genome-wide association studies, or GWAS. And so what happens in a GWAS is you get your research participant, you ask them to consent to donate DNA. Often we collect DNA through saliva. It's really easy to collect. I collect it from my samples through the post. And essentially, you then want to link, once you've got their DNA, you can genotype them using the arrays, where you're basically getting a good understanding or measuring their genetic variation at these common spots across the genome. And then you want to link that genetic variation to your trait of interest that you've also collected from your research participants, whether you've collected that from observational data, from interviews, or perhaps it's linked to data sets such as the National Pupil Database. So a GWAS is kind of like a giant statistical search party. You need lots of people, typically unrelated individuals. You collect their DNA sample, you genotype them, and then you basically test every one of those genetic variants for a statistical association with your trait of interest. The variant that's more common for people, for example, that stay in education longer versus the ones that don't, will show you that there's something going on with that variant. It's associated with education. And so the results of a GWAS are displayed in what's called a Manhattan plot. So Manhattan, thinking about the skyline of Manhattan with all these skyscrapers. And so each dot here each dot on this plot represents a single genetic variant that's been tested for association with your trait of interest. And the height of these plots, these skyscrapers, indicates kind of how big an effect size it has, so how strong that association is. These towers, in order to kind of reach genome-wide significance, these towers need to rise above a very strict significance threshold of 5 times 10 to the minus 8. And they look like towers because essentially what we're doing is, you might be testing 2 million genetic variants here, and they're ordered by chromosome, but essentially we're compressing those on the x-axis. So if you were to kind of zoom in and pull out, you will just find that these genetic variants reside next to each other or very close to each other in the genome. So this Manhattan plot, for example, is showing you that there is a region on chromosome 3 that's strongly associated with the trait being tested. So then you'd want to go off and say, what's going on in that region? What gene is there or isn't there? That might give you insights into biology. 2 key ingredients for a GWAS to be successful. You typically use unrelated individuals, and we can talk about the kind of impacts of that later, perhaps. And so they've really been facilitated again by these big biobanks, such as UK Biobank, and now the Our Future Health study, right, that's trying to collect 5 million individuals and link their genetic data to kind of health outcomes. But also the power to detect an association depends on how common the genetic variant is in the population, so how common those DNA differences are. They tend to work better when the variants are common versus rare, and how strongly they affect the trait, so what the effect size is. And GWAS they can get good at detecting very small effects, but you'll see what's needed in the coming slides to do that. So in 2014, I was very fortunate as part of my PhD to perform one of the first genome-wide association studies for receptive language ability. So here we administered, or the TEDS researchers administered, internet-based measures of receptive language, so looking at vocabulary, semantics, syntax, and pragmatics. We made this kind of overall measure of what we're calling receptive language ability. We looked in the TEDS cohort where we had these measures, and we also had genetic data. And so we tested just shy of 2 million genetic variants for association with language in this sample of TEDS 12-year-olds. And we had what we thought was a really large study, a really large GWAS of just over 2,000 12-year-olds. And you can see here, this is my Manhattan plot from the GWAS. It's not looking like the skyline of Manhattan. There are no towers. My joke is it looks much more like the skyline of Croydon. It's really quite flat. No, we didn't find any genetic variants that reached genome-wide significance, and even our kind of most significantly associated variant failed to replicate. We're scratching our heads. What's going on? We thought we were well powered enough to identify modest effect sizes. So perhaps these effect sizes are smaller than we thought. So if they're smaller than we think, then we need larger sample sizes. So we did exactly this. So cut to 2021. I'm now at Birkbeck, and my PhD student Georgie Donati with Araz Dumentel is a second supervisor. She performed 3 separate GWASs looking at educational, looking at attainment in science, looking at attainment in maths, and looking at attainment in English in adolescence. So we actually used measures from the National Curriculum SATs results at age 11, so the end of Key Stage 2, and age 14, so the end of Key Stage 3, and performed a genome-wide association for those 3 measures in the Ausbach cohort from Bristol. Again, we thought we were really nicely powered. This was a big GWAS. We had 6,000 adolescents from Ausbach. And again, from our Manhattan plot, you can see we're starting to see some towers, but essentially same story as before. This time we had 1 genome-wide significant association for science attainment, but we failed to replicate it in an independent sample. Although I will caveat with saying our replication sample was smaller, so we were making life hard for ourselves. We could also, at that time, methods have been developed and they're kind of widely used now to estimate heritability directly using genetic data or using the results of a genome-wide association study. So traditionally, if you wanted to estimate heritability, you would have to use these specialized twin cohorts and looking at the relationship between identical and non-identical twin pairs to infer a genetic effect. Analytical methods have been developed that allow us to estimate heritability using the data generated from a GWAS. And essentially what they're doing is they're measuring the degree to which the genetic variants assayed or measured in a GWAS contribute to that trait. And it's called SNP-based heritability to distinguish it from twin-based heritability. And you can see here our SNP-based heritability was actually really high. So again, in our sample, we're not finding any associations, but we kind of should be. Like our SNP-based estimates of heritability were really high, from kind of from 36% all the way up to about 50%. So what's going on? And actually around that time, what actually became apparent is that these traits we're looking at, particularly when you're looking at really complex kind of social traits such as educational attainment or educational achievement, is that the likelihood is that the effect sizes, although they're heritable, the individual effect size of any genetic variant is really small. And essentially these are highly, highly polygenic traits. So poly meaning many, genic meaning genes. And so the prediction around this time was that genome-wide association studies would be able to identify associations if the sample sizes were increased, and the larger the sample sizes you use, the more associations you will find. And so this leads me to the slide on educational attainment, or edu years. And so educational attainment is easy to measure, right? So researchers don't need access to complex school records or linkage to the National Pupil Database, for example, to get key stage results like we had for ALSPEC. Instead, you can simply ask research participants to report their highest level of education achieved. Is it a high school certificate? Is it a university degree? And this can then be converted into a years of education equivalent, meaning that researchers can basically, if they kind of collaborate, can start to perform a GWAS on very large numbers of individuals. And this is exactly what was done. A consortium was formed called the Social Science Genetic Association Consortium, and they basically performed a series of genome-wide association studies for educational attainment. And so the fourth and most recent one was published in 2022, and you can see here, this is the results. This is the Manhattan plot for that most recent GWAS. And just to indicate, because it's actually not obvious from looking at it, this here, where the arrow is the line of significance. So anything above that line is showing very strong association with educational attainment. So you can see now there's huge amounts of signal. And they did it essentially by brute force. Their GWAS was using 3 million individuals. So again, this was a big consortia effort from the Social Sciences Association Consortium. So it identified just shy of 4,000 genetic variants associated with educational attainment. The main kind of, I guess, point I want you to take home here is that no single genetic variant they identified has much of an impact. And so each genetic variant seems to contribute a very, very small amount, and at most 0.02% of the differences in attainment measured in that population. And then there's a very long tail of increasingly smaller effect sizes as you move through the list of associated variants. So it's extremely polygenic. And so there does seem to be, it is heritable, and common genetic variants play a role, but it's going to be thousands of them. So thousands of these common variants underpin the heritability of these traits that relate to education that seem really complex, such as attainment and achievement. And so that kind of left us with a bit of a problem. It's like, what are we going to do with these really small effect variants? Because one of the things I mentioned earlier is one of the reasons we wanted to perform, or researchers want to perform, a genome-wide association study is to understand mechanism. we're hoping we'll find associations that perhaps link to a gene that change how a gene functions, and that will give us an idea about biology and what's going on perhaps in the brain. But it turns out while we find these associations, most of them are in the spaces in between genes, which again makes sense because they're having a very small effect or very small impact on outcome. So they're just slightly somehow having an impact or changing how the information encoded in the DNA is accessed and used, and we're not quite sure how. So it left us with a bit of a problem that we've got these very small effect sizes for any individual variant. Also, the kind of mechanisms underpinning it are not clear at all. There's no clear kind of biological picture. What actually happened was the development of these things called polygenic indices, or you might have heard them be called to or referred to as polygenic scores or polygenic risk scores in the literature, particularly in relation to health. And so while these individual effect sizes for any genetic variant identified in the GWAS are small, and we don't know how they work necessarily, they seem to be additive. And so you can actually sum them up together to create a polygenic index. And this polygenic index can kind of summarize the genetic or the known genetic predisposition to a trait for an individual. So here, instead of looking at one variant at a time, like we're doing in a GWAS, a polygenic index takes the information from a GWAS and combines the effects of thousands of variants spread throughout the genome. And you can use this information to calculate a polygenic index for a different sample, your sample of interest, your population of interest. And in that population, you look at the variants and you weight those variants based on the strength and direction of association from a GWAS. So for example, up here on the slide, I'm showing here are the results of a GWAS, very kind of simply illustrated. I've looked at a genetic variant here and found that it's associated with educational attainment, for example, and I find that the A allele for this variant is associated with an increase of 1.5 in terms of increasing attainment. So I then look at my independent sample of individuals that I've got genotype data for. I don't have information about attainment for them, but I can calculate their polygenic score, their genetic predisposition for educational attainment using this data. So I can say, if I look at those same variants, I can count up how many of the kind of alleles they have that are associated with higher attainment or lower attainment. So here, for example, this individual has two AA alleles. Each A allele is associated with a weight of 1.5, so they get a score of 3. And you do that for all the variants that you identify in a genome-wide association study. In fact, you don't even need to, they don't even need to be kind of genome-wide associated. It turns out you can kind of slightly lower the bar and almost take all the variants from a GWAS. So you do that for thousands, up to 10,000, probably more genetic variants. And then for each of those scores you add them up to a single value. So this is the kind of the polygenic score that you've now got for each of our 4 individuals. This individual has a score of 2, this individual has a score of -2. And you can imagine now if you do that in a population of 200, 500 individuals, and you look in that population, you'll find that the polygenic index is normally distributed. So there's a few people, a few group, kind of 5% of people will have a very high polygenic index or high value. Some people will have a low. Most of us will be somewhere in the middle. And so there is a very, I guess, kind of powerful polygenic index now for years spent in education. So the Social Sciences Genetic Association Consortium did this for their GWAS of educational attainment, and they calculated a PGI from the most recent GWAS, and that polygenic index actually accounts for up to 16% of the variation in time spent in education in independent samples. So their kind of out of sample prediction is around 16%. So this is an image that I've stolen from their paper. So here is their polygenic index for educational attainment based off the GWAS data. They're looking in an independent sample. Each dot is an individual in their polygenic index. So again, if you'd kind of want to plot that as a frequency histogram, you'd see most people are in the middle with a few people at the extremes. But there is a slight correlation that on average, individuals with a higher polygenic index are staying in education longer than those with low. But there's a huge amount of scatter, right? So it's not at all accurate for predicting educational attainment for any individual, as you'd expect. But this population level variance explained makes it actually quite useful as a kind of a variable in research. So explaining or accounting for 16% in variation in educational attainment actually makes it a stronger predictor of attainment than measures of household income and marital status, and a similar strength to mother or father's education. So that's roughly about where this 2022 PGI is sitting, around about 15 to 16%. And this is just looking in different independent samples. It has been pointed out to me that although we're saying, oh, look how good it is as a predictor, 60% is massive. Another way of looking at this slide is that saying taking an average of your parents' educational attainment, which is arguably much simpler and easier to obtain, remains a more powerful predictor. So yeah, interpret that which way you like. And so polygenic indices now exist for a range of traits related to education. Most are not as kind of powerful in terms of variance or liability explained as the educational attainment, but they're getting better. So polygenic indices exist for attainment, they exist for general cognitive ability or intelligence, for dyslexia and ADHD, for your body mass index, and for diagnosable health and medical conditions such as anxiety and depression. And again, because of this kind of brute force approach of you can find more associations by conducting genome-wide association studies using ever larger samples, there's basically these PGIs continue to improve year on year as these studies continue to be conducted. So a major limitation, however, is you can kind of, the genome-wide association studies have been arguably, I think, have been a success. But the big problem is that they've only been conducted in quite homogeneous samples, and principally they've only been conducted in samples of European ancestry. So something like 94% of all genome-wide association studies to date have been performed in populations, kind of European populations, despite the fact that they only make up about 16% of the global population. That's a really big problem because actually genetic findings don't particularly port very well from one population to another. So the findings, as soon as you start looking in samples whose ancestry and kind of demographics and even kind of whether the kind of ratios of males and females start to differ from that in the original GWAS, you'll find that the performance of the polygenic index really starts to fall off. And so again, this is something that was talked a lot about in my work in the reports published by the Nuffield Council on Bioethics and Nuffield Foundation about there's real risks here, that the basic research is essentially risking perpetuating social and economic disparities in society. And I'll not dwell too long on this slide, but just to say that there is will and effort and incentives to fix this from the scientific community, with big support from funders and journals. They're taking it really seriously, but progress is really quite slow. So Our Future Health is kind of really one way to tackle that. So Our Future Health is really trying to capture much more diverse populations within the UK, and that All of Us is kind of an analogous program in the US. So while the lack of diversity in genomic data sets kind of limits the portability and the kind of predictive ability or predictive power of polygenic indices to non-European populations, they actually still can be quite a valuable research tool when you're looking in European populations. And so what I want to talk about a little bit now is the ways in which they're being used as tools in research to, I think, address questions that are important or relevant to education. And again, I've got this from from the Nuffield report that goes into many more details about the various ways that you can essentially use polygenic indices um, as research tools that might help you kind of inform practice or policy longer term, basically these kind of indirect insights. So for example, they're going to help us, or we think they're going to help us explore gene environment interaction. So we can start to ask questions like, would a high quality school environment buffer the risk associated with a lower educational attainment PGI? So there's some nice work that's come out from the Meaburn study asking just that. We can also use it to strengthen causal inference from observational data, such as using Mendelian randomization approaches. Again, there's a lot of interest in using them to improve prediction models. And then finally, what I want to actually talk about is they can be useful for accounting for confounding. Basically, they're quite useful as a way to start to untangle the gene environment correlation knot that we can kind of see within families. So I'm going to give you a concrete example of this from my lab's research that was again led by Nisa Rainey. So the naive assumption that we might go into, and I think I was probably naive to this assumption as well, is that when you're finding genetic associations, you think that you might the inclination is to think that they are causal because reverse confounding is not possible. Like your DNA sequence you're born with doesn't change during your life course, but you can still get confounding from other sources. And actually, it turns out genetic effects are not independent of environmental processes, but also vice versa for there's any social scientists out there amongst you, are probably keenly aware that environmental processes are not independent of genetic effects. And so a kind of classic example I'm giving here is when we're thinking about environmental effects. So here we're showing what can be called vertical or cultural or environmental transmission, when parental traits impact child traits through the environment they create. So the classic example is the number of books in the home having an impact on a child's reading ability. If you find this association, this might be interpreted as being a causal effect. But critically, parents share 50% of their segregating genes with their biological children. And so this path or association can actually be fully or partly accounted for by genetic confounding. Essentially, there's this backdoor path of genetic transmission whereby the genes for reading ability are being inherited by the child. So in this image here now, a child's rearing environment or home environment is influenced by the parental genes, causing the child's genes in their environment to become correlated. And we call this passive gene environment correlation. So from an environmental perspective, you've got genetic confounding going on. Now, if that child is participating in a GWAS of reading ability, the signal being detected is now not only the effect of their own genes, so via genetic transmission, the child's inherited the genes for reading ability from their parents, it can also be picking up these indirect effects of the environment. It can basically, a GWAS can be picking up the environmental effects of their genetic relatives. So from a genetic perspective, you've got environmental confounding. So how do you unpick these two things? How do you disentangle parental genetic and non-genetic influences on child development? So traditionally, all that slide text is really small, sorry. Traditionally, you would do that using twin and adoption studies. They make certain assumptions. They are specialized cohorts that are difficult to collect. And as a result, they tend to be smaller and typically not shared between researchers. And what Con colleagues realized in 2018 is that polygenic indices, in fact, the polygenic index for educational attainment, can actually be another way of using some available samples to try and unpick gene environment correlation within families. So what they did that was really quite clever is they used trios, so both parents and the child for whom genetic So basically, they've got information to make polygenic indexes. Genetic and trait data were available, and they wanted to ask if the educational attainment polygenic index was picking up the indirect effects of parental genes on child attainment that are mediated through environmental pathways. So kind of trying to see what component of the polygenic index is basically this genetic nurture effect. It's essentially environmental transmission. They did this in a really clever way. They basically divided the polygenic index for educational attainment into two separate ones for both parents. A polygenic index of the alleles shared with the child, so you've got your genetic transmission in the middle, and a polygenic index of the alleles not shared with the child. And so that's your kind of genetic nurture pathway. So this allowed them to quantify the influence the parental educational attainment PGI on the child's educational outcomes, essentially after accounting for the effect of the child's own genes. So to what extent do parental genes explain child education outcomes above and beyond the effect of the child's own genes? And what they found is about a third of the signal is coming from these indirect paths. So they're finding out about a third of this signal is mediated by genetic nurture. And when they looked at kind of candidate mechanisms, or some of these things seemed to be mediated by characteristics such as family socioeconomic status. And there's a really nice, it's very small, a very nice kind of meta-analysis that was published a few years ago from a group in UCL that then kind of shows that this genetic nurture effect for educational attainment is really very robust and replicable. You might be saying, well, why does it matter? I think it really matters. Essentially, understanding these kinds of paths within families helps us kind of understand if we want to help our children kind of succeed, we need to think about where we're going to focus our efforts. And we can hopefully obtain less biased estimates of both genetic and environmental effects. So if we can actually isolate direct genetic effects from the biology of a child, that might be closer to biological mechanisms, perhaps. Again, we haven't been super successful in mapping genetic signals to mechanisms. If we can identify genetic nurture effects, we can then think about kind of quantifying these effects and identifying which parental behaviours and rearing environments might mediate those. And this might be giving us targets for intervention, or at least give us an idea of effect sizes we might expect if we were to intervene on these pathways or processes. And we can also think about, when these effects are fold across development. So I just want to lead us into the genetic nurture study that Nisa in my lab led along with colleagues at UCL and in the Netherlands. And so we wanted to ask, how do parents' genes affect their child's risk for psychopathology? Psychopathology broadly meaning kind of mental health. Is it through direct genetic transmission, or is it through this environmentally mediated genetic nurture? And so we looked at internalizing and externalizing behaviors in adolescence. So we looked at age 11. So internalizing behaviors, these kind of inward behaviors such as anxiety, social withdrawal and depression, or the kind of outwards externalizing behaviors of aggression, impulsivity and conduct problems. These things we know, both internalizing and externalizing, are highly heritable, and they link to adverse educational and social outcomes and things like employment. Critically, we also know that parental involvement and parenting strategies are heritable, and they also link to child mental health. And so it makes it a really good candidate mechanism for genetic nurture. So we performed a genetic nurture study, kind of following in the footsteps of Kong et al., where we wanted to ask whether parental genetic liability affects child internalizing and externalizing traits through genetic transmission and through indirect genetic nurture. And second, if we found genetic nurture, can we identify any specific parenting behaviors that mediated this effect? And so this is Nisa here, my student. So this is our study design. And so we basically we didn't quite follow the same approach as Kong et al. We used the statistical control method, which is basically using a path model to statistically separate out the effect of direct genetic transmission from this indirect genetic nurture effect. And so we used data from just under 800 parent-child trios from a Dutch longitudinal study called TRAILS, the Tracking Adolescents Individual Lives Survey. And our genetic measure, our polygenic index we used was for general psychopathology. So here we wanted to take a transdiagnostic approach and essentially try and capture genetic liability for mental health in general. So capturing genetic liability that's shared across 11 different psychiatric conditions. Our child behaviour, we were looking at internalising and externalising as reported by the parents. And then we had these candidate mediating mechanisms. So again, looking at these, if we'd find genetic nurture paths on the outside here, we wanted to ask if these would be mediated by parenting. Particularly, we wanted to look at parenting, parental warmth, overprotection, or rejection. And these were child reports of parenting, so child perceptions of parenting. So what did we find? Well, first of all, the sanity check was that do we see what we'd expect to see with this polygenic index in our sample? So this is not showing the path models. This is now just showing kind of pairwise correlations between the children and their internalizing, parental rated internalizing and externalizing. And thankfully, as expected, our polygenic index, the child's polygenic index, does in fact associate with their internalizing and externalizing behaviors in the direction we'd expect. Big few, that was a kind of big sanity check. And also both the mother and father polygenic index, their own polygenic index for psychopathology, correlated with their child report of externalizing and internalizing, although that only came from the mother. But again, that kind of makes sense in terms of what we're expecting. What we failed to find, however, is any kind of candidate mediating mechanism. So we didn't find any associations between our parental polygenic indices and child reports of their respective parenting. So that kind of ruled out our planned genetic nurture mediation analysis because we didn't actually have that basic association between parental genetics and their parenting. So the main findings, moving then on to the kind of the path model, we didn't find any evidence of genetic nurture at all for externalizing problems, but we did find it for child's internalizing problems. And in fact, what we found is that the mother's polygenic index for psychopathology is linked to child's internalizing problems, but it seemed to work almost entirely through this environmental pathway of genetic nurture, so this bar on the left here, rather than through direct genetic inheritance. Somewhat surprisingly, we didn't find genetic nurture effects for internalizing from the father. It's just coming from the mother. And also, more surprising still, that caused me to scratch my head for some time, is we actually no longer find direct effects of the child's polygenic index on their internalizing, despite the fact that when we're doing pairwise correlations, essentially doing a between study correlation, we do find quite strong effects, quite a strong relationship between a child's polygenic index and the child's parental report of their internalizing. You'll also note that there's some evidence that the parental polygenic scores are correlated, which is possibly indicative of assortative mating between the parents. So our kind of questions and interpretations. So we didn't find genetic nurture effects for fathers. This might be because mothers are often the primary caregivers who shape the early prenatal and postnatal environment for their children. That's one kind of candidate explanation. Or it could also just be a bit of a confound or bias in the reporting or measurement of how we've measured kind of child internalizing. So when we looked at who completed the reports, it was always largely the mother. So there may be a kind of a bias, potentially genetically influenced bias there around how mothers are reporting their child's own internalizing and externalizing behaviors. We weren't able to answer the question of how maternal genetic nurture works because we basically probably weren't looking at the right candidate mechanisms that were available to us, parental warmth and rejection, because they weren't associated in the sample. And then we didn't find any genetic nurture effects either from the mother or father from externalizing. That goes a little bit against what other research has been finding. But again, this is quite new and the picture's quite mixed. But it might be that perhaps we're looking at the wrong age. Perhaps age 11 is too young to find these effects. And finally, I think, again, this really blew my mind for a while. Essentially, we didn't really find direct genetic effects once we account for the effects of the child's, the genetic effects of the child's parents. Essentially, this is telling me that direct genetic effects are more complex than we're expecting. Again, one possible explanation is that kind of the environment the parents create might be obscuring or overriding the expression of a child's own genetic predisposition. That's one kind of possible explanation. Or it could be that the polygenic index we're using is really, it's comprised of social and environmental effects, and it's not actually picking up direct effects of a child's biology. So future directions that NICE is currently hard at work on at the moment, and my other student Hattie's also looking at, is fundamentally, I think we need richer environmental measures to pinpoint specific genetic nurture mechanisms. And one thing we have available to us are direct observations of parent-child interactions that NICE is working with. I think if we want to understand how genetic and environmental effects unfold across development, we really need these studies that track families over time. So using longitudinal studies, again, we have some of those available to us here at Birkbeck. And we took this kind of transdiagnostic approach to mental health by looking at this general psychopathology factor, not a specific one. And again, we want to now kind of ask similar questions, but looking at more specific polygenic indices, so that those linked to ADHD and autism. And again, as mentioned, essentially we're very limited here. We need bigger and more diverse samples. Again, we're looking in European ancestry samples that are very homogeneous, which makes it difficult to kind of generalize these findings outside of these groups. So if I've got time, where am I doing for time? Oh, if I've got time, quite quickly. I've just been talking about kind of basic research that longer term might indirectly inform policies focused on understanding transmission of educational disadvantage or mental health within families. But I just want to touch briefly on the idea of, can genetic data or polygenic indices actually have direct uses in education? So can they kind of be applied in ways if we can kind of handle, collect and interpret the data responsibly? And I think this is a question we need to ask now, because educational genomic research isn't happening in a vacuum, right? We're following in the wake of the enormous effort that is health genomics. So all the infrastructure, the public debate and knowledge exchange and the technology that's being developed for health is inevitably going to pull in other fields like education along with it. And I think it makes it really important that we start having conversations now about what the science can and can't tell us, and to think more deeply about the state of translation in relation to education. So just to kind of highlight here, right, that the UK government's Genome UK strategy is a 10-year plan to become the world leader in genomic data. We've got the Our Future Health study that's aiming to link genetic data to health and lifestyle data for 5 million people. There's huge amounts of kind of effort and interest. And so this raises the question, or the question has been raised, particularly in Robert Plomin's book, is that can you use genetic information directly? So can you essentially genotype children and calculate their polygenic indices and use these as tools in society, potentially mirroring those applications that are being discussed and being tested in healthcare? So for example, can you use them as population level screening to guide further action? Can you use them to support early identification of educational needs? So the idea is you can kind of step in and help individuals and their families earlier. perhaps they might inform the personalisation of educational provision, kind of, I guess, allied to the idea of personalised medicine. And it's worth reflecting on why there is interest, right? What sets polygenic indices apart from other early markers of educational difficulties? Why wouldn't you just use other more accessible predictors that are less ethically fraught, or the actual trait you're interested in? Just measure the actual trait you're interested in, such as realised attainment. So I've just kind of got, I guess, my kind of interpretation of what the possible advantages or opportunities are for using polygenic indices in a kind of in the real world context. Number one, the kind of predictive power of some of these polygenic indices, particularly attainment, means that you can start to improve prediction metrics, particularly when they're combined with other measures. Your polygenic indices are fixed from birth and stable across development. And because they're specific to the individual, they can differentiate between children in the same family in the way that family level variables such as social disadvantage can't. Not everyone knows their family history, so sometimes the variable you're interested in is unobservable or unavailable. In theory, you can obtain a PGI data for everyone. You don't need a clinician or a healthcare to get it. You don't necessarily need your family history. But of course, those characteristics also make the ethical landscape of genomic prediction fundamentally different from other markers or predictors of risk and resilience in education. And so just to say, and again, I suspect Michael Thomas will talk about this more in his seminar, this has not escaped the attention of policymakers. So the Department for Education are starting to ask kind of, can we use this information, particularly in relation to educational need? And so they've published 2 reports kind of specifically on genomics. The most recent one was last year when they focused on 2 potential applications of genomics in relation to the commercial market and in relation to screening at birth, special educational needs and difficulties. And then in their areas of research interest that they published in April this year, again, they mentioned genomics directly in relation to SEND. And so I think this is nearly my last slide. And so it's worth thinking about, I've talked about potential advantages, but also thinking about limitations. And again, I'm thinking about limitations of these polygenic indices in relation to education. This might not necessarily hold for medical or health conditions. They're sample specific, right? They perform poorly for non-European populations. They're not accurate for individuals. I haven't mentioned this much, but essentially they're non-specific. The polygenic index for educational attainment also correlates with health conditions. It correlates with psychopathology. Doesn't tend to be have specific effects. For the most part, it doesn't tell you anything about mechanism, what these variants do, which means we don't necessarily know how to act or how to change outcomes. And as I've shown, we know they're picking up environmental confounding, so they're picking up the effects of genetic relatives. And so that might mean you're acting on information such as social disadvantage within families. And so in spring this year, we started the Centre of Educational Centre of Educational Neuroscience Genomics Working Group, so CENGEN Working Group, to basically think more deeply about genomics and education. And an initial goal of this working group is to map out our position, certainly, or the centre's position on translation, which we hope will be useful to educators and to policymakers. So thinking about the state of the science and state of translation, but really also to set out, which I wasn't able to do before, set out some blue sky thinking about what is needed for research and research infrastructure to enable evidence-based translation. So here are the colleagues in the working group from Birkbeck, but also from UCL, from IOE, and from King's College London. So watch this space. And thank you to my colleagues and collaborators. I'm out of breath. I've talked very fast. Thank you. I take questions.
Superb. Thanks so much, Emma. Fantastic tour through all the technicalities of the area. We're about at 5 o'clock. We'll just take maybe 5 minutes extra, if that's okay with everyone, just to ask some of the questions that have come up in the chat. So Jared asks, I'd love to know what percentage of general cognitive function is explained by PGIs. Is that similar to educational attainment level?
It's about 5%. So it's about 5%, but all interestingly, so there is a GWAS for intelligence, but the educational attainment polygenic index is better at explaining variance in intelligence than the intelligence polygenic index. And this is going off topic slightly, but if you start to bundle these different polygenic indices together, they're partly non-overlapping, so you can start to build more predictive models. So people are taking this, what they're calling multi-polygenic score approach, where they start to kind of add these together to try and, yeah, explain more variance. But you're around I think 5%, just looking at one polygenic score.
Even though cognitive ability is more highly heritable than educational attainment, is that just a sample size thing?
Hard to know. The sample size is quite, yeah, the sample size is smaller. Yeah.
No, it could be that. Yes, yes.
Yeah, potentially.
We always need bigger samples. That's what they used to say about twin studies. If only we had bigger samples. Booth J asks, excellent talk. Is there evidence for genetic nurture effects for language ability?
Oh, I've printed it out because I didn't have time to read it. That's a really good question. There is, if I find it, maybe ask me the next. There is a recent paper that was published by Saloni. I'll have a look. Not that I know off the top of my head, but I know there was one about reading. It might have talked about language, genetic nurture effects. If I can find it, I can ping it.
Okay, yeah, send it to me because
Yeah,
I'm going to be talking about this stuff in a couple of weeks. In fact, I'm really excited to hear about the talk I'm giving from you, Emma.
I'm just, yeah.
All right, Catherine says, unfortunately, markers such as socioeconomic status are well identified as putting children at risk for education. And even when we have such obvious markers, we do not find the right key to help those children. So
Yeah.
I wonder how genes could help us further.
Oh, yeah. I mean, yeah. Fundamentally, I think you're fiddling around the edges until you kind of address some basic social problems in society. You know, it's not going to have enough. You need to kind of make sure all the children are arriving to school well fed, with good access to education first and foremost before you start thinking about genetics, honestly.
Very good. Sam says, could you foresee that you might come to the conclusion that genetics is not actually that useful for education?
Yeah. As a geneticist, I mean, yes and no. I think it's I'm yet to be convinced about kind of translational potential. I guess this is kind of why we've got the working group, right, to really think a bit harder about these issues. I think it is really important, though, that we have more interdisciplinary research. So really thinking about this gene environment correlation so that you're not going to be in a situation where you've got one group of people saying, well, genes are causing this, and one group of people saying environment's causing this, that you have a much better understanding. So I think that will be longer term research wise, and it requires much better collaboration in interdisciplinary research.
Super. And I will just have the last question as my privilege. Oh, no, there's one more coming. I've got to ask this. What is the percentage of role genetics plays in the development of a child's learning as compared to their circumstances?
Well, so I guess the rough answer to that is 50-50, right? So when you're looking at, heritability, it seems about 50% of the differences are due to genes. 50% is environment, and then you can kind of split that environment into shared and non-shared effects. But they tend to, when you're looking at education, they tend to be effects that differ between families, so non-shared.
Okay, Gabrielle, we've got some late breaking questions.
Yeah.
What do you make of the fact, Gabrielle says, that most of the SNPs identified are in the introns? Does this indicate a flaw in the methods, or does it give us clues as to the role of introns? So it's just non-coding bits of DNA.
Yeah. It's just where I mean, that's why it's kind of just where we're looking. And so we're missing, when we're doing these association studies, we're missing a really big part of the picture. And that's why when you're estimating heritability from genetic data, it's nowhere near, or it's typically half that you'd see in twin studies, because there are going to be other variants, rarer variants that are involved, but they're just harder to find. But those are going to be much more likely to be in genes. So we're just getting a bit of the picture. I don't think we can read too much into it. I think it's a matter of where we're looking.
Great. And my finishing question, talked a lot about genomics down in the DNA, but people may have heard about epigenetics. Is that something much more important we should be worrying about? Or are we interested in that? Or do we not have to worry about that?
Yeah. Epigenetics can be very, very zeitgeisty. It can be very persuasive. I would, epigenetics is just, so for those of people who are not familiar with epigenetics, this is essentially this kind of layer of chemical information that sits on top of the genome that regulates how the information encoded in the DNA is accessed and used. Importantly, epigenetic changes are also heritable. So some of the epigenetic differences you'll see are basically, they're the mediating mechanism of DNA sequence, right? That's how the DNA sequence is having a change. Some of them they're also kind of stochastic, a bit random, but they do seem to be changed systematically in relation to the environment. The problem we have is that it's very, very hard to unpick those signals in human samples, because your epigenetic profile changes between individuals, but also even within an individual across their different cell types and across their life course. And so most of the research, when you're looking at kind of epigenetic changes in relation to these complex things, you're limited to animal studies where you can fix the genome, fix the DNA sequence, and manipulate the environment. So I would take epigenetic studies in relation to educational attainment with a big pinch of salt. What's the word I want to say? With a kind of, yeah. Put your critical thinking hat on.
Yes. Okay. But I mean, they're interesting because they're environmental effects that can go across generations. But
careful with generations there.
Well, yeah, maybe why. But anyway, I mean, I would probably add into that if you're looking at epigenetics, you're getting into the mechanisms of how environments influence things rather than just, so it's on the environment side, getting into the mechanism.
Yeah.
All right. So is that okay to someone?
Yep. That's
good. Okay. So I'm gonna thank Emma so much for a fantastic talk. Thanks for everyone for staying with us and some fantastic questions. Next week, we have a seminar by Professor Arnold Wilkins, who'll be talking to us about visual stress and how that might have educational impact. And then in 2 weeks' time, Emma has set things up very nicely for me, where I'll be talking about the sort of translational aspects of genomics. How might you actually use genetic information in a way that will be relevant to practitioners, educators, and policymakers? Until then, thanks very much, everyone.
Thank you. I'm just reading my questions.
Lots of good ones.
Michael, you might have to switch the recording off. I seem to have lost my button to control it because I